Welcome everyone. My name is Alazar Tadesse. I am grade 12 biology teacher. I'm happy to take you through Unit 2 microorganisms, a fascinating world that's invisible to our naked eye, that's very essential to life as we all know. In this session, we'll explore a wide range of topics, including Eubacteria, bacterial structure and reproduction, their genetic exchange, and the incredible archaea that live in extreme environments. This educational video is brought to you in collaboration with Bright Technotonic Company, supporting quality science educations through innovative technology. Get ready to explore how microorganisms shape our world. Let's begin. Unit 2 is about microorganisms. Microorganisms are tiny life forms, meaning they are very small. They are too small to be seen with our naked eye. Naked means bare or without use of supportive instruments like microscopes or magnifying glass over our eye. We cannot see them. So they can only be seen with the aid of what? Instruments that can enlarge their size. Since they are very small in their size, they need to be, or we need to use what? Instruments that can magnify or increase their size in our eyes. So they cannot be seen without the aid of those instruments. Because of this, they are known as microorganisms. Here there is another important term, microbiology. Microbiology is a science, or it's a study of these microorganisms or tiny organisms, which includes the bacteria, the viruses, the protozoa, the fungi, the algae, the parasitic worms or the helminths. In this presentation, we'll explore the fascinating world of microorganisms, focusing on the two major domains. By the way, there are about 3 domains of life. Out of the 3 domains, today we will focus on the 2 domains of life. The 3 domains are the bacteria or the Eubacteria, the archaea, and the Eukarya. The Eukarya includes those eukaryotic organisms. That means, in another word, the remaining microorganisms and other organisms, the multicellular organisms, are included under what? The animals, plants, and those remaining microorganisms other than the bacteria and the archaea are included under Eukarya. So the Eukarya group will not be discussed in this session, but the 2 major domains will be discussed. We'll examine their structure. So while discussing about these 2 domains, we'll focus on their structure, on the reproductive matrix or on the reproduction matrix, and the diverse environments they inhabit, from everyday surroundings to the extreme conditions that will be lethal to most life forms. So we will focus on these major areas. Let's begin with the first one, Eubacteria. Eubacteria are known as true bacteria. They have the following characteristics. They are unicellular, means their body is composed of a single cell. They are made up of only a single cell. And they are prokaryotic, meaning they don't have true nucleus, or they lack nuclear membrane and other membrane-bounded organelles like the Golgi apparatus, mitochondria, and chloroplast, plastids, and the likes are not present in their cellular structure. Because of this, they are sometimes known as simple organisms because of their cellular structure. The other important characteristics of these bacteria is they have thick peptidoglycan cellule. They have a cellule, and the cellule is made up of or composed of what? Peptidoglycan. A peptidoglycan, which is composed of the different substance that makes up substance that's different from that makes up the cellulose of the plant cellule, the peptidoglycan. They have somehow thick peptidoglycan cellule. So it has distinctive cellular composition that provides structural support for the bacteria. The other important characteristics of these Eubacteria are they have varied nutritional modes. Some can be autotrophic, meaning they can make their own food, while some others can be heterotrophs. That means they consume organic matter. The other characteristics of these Eubacteria is that they lack true chlorophyll. Unlike those plant cells, the cell of bacteria lacks true chlorophyll. Rather, they have some kind of chlorophyll which is known as bacteriochlorophyll. bacterial morphology and classification. So what shape do bacteria have? So they will focus on their morphology, and we'll try to classify bacteria based on differential staining. Let's focus on the first one, on the bacterial shape. Bacteria have different shapes. Some have spherical shape or round in shape, and they are known as cocci if there are many. The plural form, cocci. And if we are talking about a single round-shaped bacteria, we can say coccus. Coccus. C-O-C-C-U-S. Coccus. Or if there are many, we can say cocci, as it's written there. The second group of bacteria have rod shape, a shape that resembles stick. Sometimes you might have heard people saying, especially in the religious arena, they may say that, or you can find this story in the Bible, the rods of Moses, the rod or the stick that Moses used to divide the Red Sea after receiving command from God. So rod means stick. So such bacteria have what kind of shape? They have a shape that resembles a stick, and because of this, they are known as bacilli, rod-shaped bacteria, bacilli. Here we have, this one is what? It shows us the bacilli or the rod-shaped bacteria. With the one which is circle in shape are what? They are cocci, okay? Cocci. The other groups are the spirochetes. The spirochetes usually they have what? Corkscrew shapes. The device or the instrument that's used to produce a pore or a hole in wood. The device that's known as corkscrew. Screw. So they have what kind of shape? The screw shape. They are known as spiral-shaped bacteria. They are spiral in shape. የእንጨት መቦርቦሪያ ወይም መሰርሰሪያ የሚመስል ቅርጽ ያላቸው ናቸው። እንጨት መሰርሰሪያ ምናልባት እንጨት ቤት ሄዳችሁ አይታችሁ ይሆናል ምን አይነት ቅርጽ እንዳለው ማሽኑ እና እንደ አይነት ሼፕ ያለው ቅርጽ ያለው ባክቴሪያ ምን ይባላል? ስፒሮኬትስ ይባላል። ወይም ምን አይነት ቅርጽ አለው? ስፓይራል ሼፕር ናቸው ይባላሉ ማለት። ሌሎችም አይነቶች አሉ። ለምሳሌ ኮማ ሼፕድ ይሆናሉ። የኮማ ቅርጽ ያላቸው they are known as vivero. ለምሳሌ ኮሌራን ኮዝ የሚያደርገው ቪቪሮ ኮሌሬ ተብሎ የሚጠራው ኮማ ሼፕድ ባክቴሪያ ነው። ግራም ስቴኒንግ ላይ ስንመጣ ክላሲፊኬሽኑ ላይ እንመጣው። now we are moving to the second portion here, the grams staining. We can classify bacteria based on their cellular compositions and based on their the staining capacity, or based on their retaining ability, or based on their ability to retain the grams stain. So these two groups of bacteria are what? Gram-positive bacteria and gram-negative bacteria. Such classification is based on whether do they retain the grams stain or do they do not retain the grams stain. If they retain the grams stain after the grams staining technique procedure, they are known as grams positive. By the way, the staining procedure involves about 5 steps. The first step is to fix the bacteria on the slide, slide in the laboratory. So first we need to attach the bacteria on the slide. We need to add the bacterial group on the slide. Then we need to add the primary stain, which is known as the grams stain, which is known as, in another word, the crystal violet stain, which has what kind of color? Purple color it has. So we add the purple colored stain, the dye, the primary stain. So after adding this stain, we need to add a mordant, which is known as iodine, which has a capability to fix the color into the, or which can help the color or the primary stain to penetrate into the cell of the bacteria, thereby helping it to get attached to the cell. So after this step, the fourth step is what? To add decolorizing agent, which is known as alcohol. Alcohol can be added, okay, which has a decolorizing character, a decolorizing function. That means it can help to decolorize or to remove the stain. If the stain is not attached firmly to that of the cell of the bacteria, especially to the peptidoglycan, it can be easily degraded or it can be easily removed. So it's because of this, the alcohol is known as the decolorizing agent. So the fourth step is to add the decolorizing agent. After decolorizing agent is added, those bacteria with thinner cellular composition, especially with a thinner peptidoglycan layer, are not able to retain the primary stain. They easily lose it. So when they lose it, we add another dye, counter stain, which is known as safranin, which has a red color. Such counter stain is, counter means ምትክ ነው በዛኛው የምንጨምረው ማለት ነው። ያኛውን ቀለም ስላጣው ባክቴሪያው አልኮሉ ሲጨመር አሁን ምን እንጨምርበታለን ማለት ነው? ሳፍራኒን የተባለ ቀይ ቀለም ያለውን coloring agent እንጨምራለን ማለት ነው። ዳይ ወይም ስቴን እንጨምራለን ማለት ነው። So upon adding the counter stain, then now this bacteria will have what kind of color? Red color, okay? Red color. So such bacteria are known as by the gram negative. They are not able to retain the gram stain, the primary stain. Because of this, they are now negative, gram negative, okay? Don't forget. While those with the thick peptidoglycan layer, or with the cellular, which is able to retain the primary stain upon the addition of the decolorizing agent, and finally appearing blue in color, they are known as what? Gram positive. So they retain the purple stain and they have thick cellular, don't forget. The gram negative, in addition to these characteristics, they have outer membrane, which is made of lipopolysaccharides and lipoproteins. They have additional membrane. Other than the cellular, they have outer membrane, okay? They have outer membrane. And usually they are known to cause disease-causing bacteria. They are usually, most of those bacteria that are known to cause disease are gram-negative. But this does not mean that all bacterial cause diseases are caused by gram-negative. It does not mean that like that. There are some gram-positive bacteria that are known to cause human disease, okay? For example, tuberculosis is caused by such gram-positive bacteria. So gram-positive bacteria also cause disease, okay? Don't forget. This fundamental technique helps to differentiate bacteria based on what? Their cellular composition, which affects their susceptibility to the antibiotics. Bacterial nutrition and metabolism. Now, the other important point that we need to address in this session is about the bacterial nutrition and their metabolism. Bacteria have evolved many mechanisms to obtain nutrients and energy so as to grow and reproduce, to multiply. They have evolved what? Many mechanisms of obtaining nutrients and energy. So these mechanisms are usually known as bacterial nutrition mechanisms, okay? So bacteria can be classified mainly into two groups, autotrophs and what? Heterotrophs, based on the source of the carbon they use based on the source of carbon they use either to make their own organic food or to obtain energy, the energy they need for their growth and reproduction. From the organic food, they may obtain the carbon either from the inorganic source, like from carbon dioxide, or either from the pre-prepared organic materials, like from glucose. Or if they obtain the carbon which is necessary to make the organic compounds. By the way, when we say organic compound, a compound must contain both carbon and hydrogen. And it may contain other elements. This is not mandatory. Other elements could not be mandatory, but in order for a compound to be organic, it must contain carbon and hydrogen. That's when we say organic compound. For example, glucose is organic. Why? Because it contains both carbon and hydrogen. So in order to make the organic compound, such bacteria obtain carbon from inorganic source. The inorganic source it's opposite to that of organic. So if organic is a compound that contains both carbon and hydrogen, the inorganic one is the one that contains either carbon or hydrogen, or may not contain both. So in such case, we call it inorganic. So the carbon from inorganic means like carbon dioxide is inorganic compound. It does not contain hydrogen, so it's inorganic. So based on this, we can say that the bacteria could be autotrophs and heterotrophs. Troph means feeding relationship. So they obtain the carbon from inorganic source, and because of this inorganic source, and they are known as autotrophs. While the bacteria that obtain the carbon from the organic source, like from the pre-prepared food, like us. We cannot make organic compounds like glucose by our own cellular mechanisms, like through photosynthesis. We cannot do this. Because of this, we are known as what? Heterotrophs. We can obtain only carbon from what? From pre-prepared organic molecules, from plants or from other photosynthetic organisms. Because of this, the carbon is brought from where? From organic source, from the source which contains carbon and hydrogen, like from glucose. Because of this, these such bacterial groups in this case are known as heterotrophs, okay? Heterotrophs. So, based on, again, their source of energy, we can classify bacteria into two trophs. They are feeding relationship, like chemotrophs and phototrophs. Phototrophs are those bacteria that obtain their energy from what? Sunlight, from sunlight source. Their source of energy is what? Sunlight. For example, just like those bacteria, green plants use chlorophyll to absorb what? Sunlight, to use the energy to what? You will learn about this in Unit 3, to obtain the energy that's needed to cause water to be lysed or to split the water, which can be act as a source of electron. So the source of energy for those bacteria, for these bacteria, is what? Light energy. And because of this, what kind of feeding relationship they have? They are known as phototrophs. We can combine the phototroph with what? The autotroph. So those bacteria with source of energy from the sun and the carbon source from the inorganic compounds like from carbon dioxide are known as what? Combine it. Photoautotrophs. We call it photoautotrophs. While the bacteria may obtain the carbon from inorganic sources, like from carbon dioxide, but the energy source could be what? Not from the sunlight, but from chemical oxidation of compounds, either from oxidation of inorganic compounds or oxidation of inorganic compounds. Such bacteria have what kind of relation? They are chemotrophs, okay, chemotrophs. And you can combine the chemotrophs, their source of energy from oxidation of chemical compounds, and their source of carbon is from inorganic carbon source, which is from carbon dioxide. They are known as chemoautotrophs, okay, chemoautotrophs. So we have two types of autotrophs here, photoautotrophs and chemoautotrophs. While the other groups have their source of carbon is what? Is from organic molecules, like from carbon dioxide, from glucose. Such groups of bacteria are heterotrophs, and if they obtain the energy that needed to, for example, for their process of respiration, is the energy is obtained from, for example, from the breaking down of glucose during the respiration process. If the energy is obtained from organic molecules or from organic carbon source, like from glucose, then such groups are known as, they are heterotrophs. And if the energy is obtained from chemical oxidation process, they are chemoheterotrophs, okay, chemoheterotrophs from chemical oxidation process. Some groups of bacteria are able to obtain energy from sunlight, but still use carbon source glucose. In such case, they are known as photoheterotrophs, okay, photoheterotrophs. When you see their environmental adaptability, bacteria can be found everywhere. Their group, bacteria can be found in the soil, in the air, in the water, everywhere you can find bacteria. So bacteria, even though one bacteria cannot be found in everywhere, but different groups of bacteria can be found in different parts of the environment. Even within your body, in your skin, there are bacteria. So bacteria can be found everywhere, we can say. So due to their diverse metabolic capabilities and adaptability to their different nutrient sources, they can be found everywhere, okay? Everywhere. Let's move to the next important point, bacterial reproduction and genetic exchange. How do bacteria reproduce? Or how do bacteria get multiplied? Or how do bacteria increase in their number? Reproduction is very important. So one of the life characteristics that living things have is what? Reproduction. There are several life characteristics, you know. like metabolism, growth, movement, irritability. There are 7 characteristics. Out of those, the 7 life characteristics, the bacterial reproduction is very important to ensure that organism can survive in the future. So since a single organism cannot live forever, so it needs to what? To replace itself. So it is through the process of reproduction. So now the second important point is genetic exchange. Genetic exchange, bacteria's reproduction does not result in genetic diversity. Bacteria have evolved a mechanism to bring what? A genetic diversity through another mechanism, not through the reproduction pathway, but through another mechanism. They have brought genetic diversifying mechanism. That's genetic exchange. But first, let's see the reproduction method or mechanism of bacteria. Bacteria reproduce mainly through what? Through binary fission. Fission means splitting. Binary means into 2. So bacteria, after growing for a certain period of time and reaching a certain level of growth, especially after doubling its mass, the bacteria's chromosome gets duplicated. Its cytoplasmic contents or organelles gets duplicated. And finally, in the middle, there is a development of what? A wall, a septum. The septum then divides the bacteria into 2. Finally, the bacteria splits into what? Into 2 identical daughter cells. Here, there is no difference between the bacteria, with the daughter bacterial cells and the original one. But the difference between those bacteria can be brought only through the genetic exchange mechanisms, like through what? Through conjugation, through transduction, and transformation mechanisms. Conjugation, it is a mechanism by which bacteria can obtain genetic diversity. It is when 2 bacteria conjugate or 2 bacteria collide with one another. They physically contact with one another, and through the help of a pilus, a structure which is found the outside of the bacterial cell. There are structures that are found outside of the bacterial cell, like the pilus, small structure that helps the bacteria to attach to a certain surface, which is pilus. We call it pili or pilus, singular pilus. There are other structures that help in motility or in the movement of the bacterial cell, like flagella. That here, through the help of the pilus, one bacteria gets attached to another bacteria. Such pilus is usually known as sex pilus. Finally, the plasmid, which is extra chromosomal material found in the bacterial cytoplasm, gets divided and gets copied and moves to what? To another bacteria. Such bacteria is known as donor bacteria, okay? Donor bacteria. The one receiving one is the recipient bacteria. So finally, the one which has received the new plasmid can have what? New characteristics. Because of what? The new genetic material it has acquired from the another bacteria. So genetic diversity can be brought through such mechanism. So such mechanism of getting genetic exchange is known as conjugation, through physical contact. 2 bacteria get in contact. Physically, they contact, and for one bacteria acts as a donor. It provides acts as a donor to provide its copy of its plasmid to the other bacteria. The other bacteria receives the plasmid, and finally, the bacteria could have a different kind of characteristics that it had. For example, the bacteria may become what? Resistant to antibiotics. It may become resistant to antibiotics. Some diseases could not be easily cured because of what? Acquiring of resistance through those bacteria, through such mechanisms. ስለዚህ አንዳንድ ጊዜ በሽታዎች በቀላሉ መታከም የማይችሉበት ሁኔታ ይመጣል። ለምን? ባክቴሪያዎች ያ መድኃኒት ከዚህ በፊት ይገላቸው ነበር። ነገር ግን በእንደዚህ አይነት ፕሮሰስ ከሆነ ባክቴሪያ ዝርያ አዲስ ጄኔቲክ ማቴሪያል በሚያገኝበት ሰዓት ምን ያገኛል? ያንን መድኃኒት እንዳይጎዳው ሊያደርግ የሚችል ሜካኒዝም ወይም ስትራክቸር ዴቨሎፕ ማድረግ የሚያስችል ጄኔቲክ ማቴሪያል ከዛ ከሌላኛው ባክቴሪያ በሚቀበልበት ሰዓት ሬዚስታንት ይሆናል ማለት ነው። መድኃኒቱ እንዳይጎዳው ይሆናል። ስለዚህ ለዚህ ያግዘዋል ማለት ነው። So direct transfer between 2 bacteria via physical contact using a sex pilus is known as conjugation. Transduction. Transduction is the transfer of DNA from one bacteria to another, but it's not direct. Rather, it is through involvement of a mediator. There is an involvement of a virus. The virus acts as what? A vector. It is through the virus that the genetic material, especially the plasmid from one bacteria, can be transferred to another bacteria. So there is no direct transfer. Rather, there is involvement of a virus. Especially what kind of virus? Viruses are known as bacteriophage. Why they are known as bacteriophage? Phage means virus. Bacteria means those viruses that attack bacteria. They are known as bacteriophage. So sometimes some bacteria attack one bacteria, and after attacking that bacteria, they what? They cause that bacteria to multiply their genetic material. እነዚህ ቫይረሶች ባክቴሪያዎችን አታክ አደረጉ በኋላ የራሳቸውን ጄኔቲክ ማቴሪያል ወደዛ ባክቴሪያ ያስገቡና የነሱ ጄኔቲክ ማቴሪያል እንዲባዛ ውስጥ ለክ ሪፕሊኬት እንዲያደርግ ያደርጋል። በዛ ሂደት ውስጥ የባክቴሪያው ጄኔቲክ ማቴሪያል ከቫይረሱ ጋር ሊቀላቀል ይችላል ማለት ነው። ኢንተግሬሽን ሊፈጠር ይችላል። ከዛ በኋላ ያ ቫይረስ ያንን ባክቴሪያ ከገደለ በኋላ ወጥቶ የራሱን ጄኔቲክ ማቴሪያል ይዞ ሌላ ባክቴሪያ አታክ በሚያደርግበት ሰዓት የራሱን ጄኔቲክ ማቴሪያል ወደዛ ባክቴሪያ ሲያስገባ ከዛኛው ባክቴሪያ ያመጣውን ጄኔቲክ ማቴሪያል ምን ሊያደርግ ይችላል? ወደ ቀጣዩ ባክቴሪያ ሊያስተላልፍ ይችላል። በዚህ መንገድ ያኛው ባክቴሪያ ይሄንን አዲስ ጄኔቲክ ማቴሪያል ካገኘ ጄኔቲክ ኤክስቼንጅ ከተፈጠረ ትራንስዳክሽን እንላለን ማለት ነው። ሌላው ትራንስፎርሜሽን። በዚህ መንገድም ጄኔቲክ ማቴሪያል አኳየር ሊያደርጉ ይችላሉ። ጄኔቲክ ዳይቨርሲቲን ሪሲቭ ሊያደርጉ ይችላሉ ማለት ነው ባክቴሪያዎች። ይሄ ትራንስፎርሜሽን ይባላል። ይሄ ደግሞ ከየት ነው? በሁለት ባክቴሪያዎች መካከል ሳይሆን በባክቴሪያና ኢንቫይሮንመንት ካለ ጄኔቲክ ማቴሪያል ነው የሚሆነው። አንዳንድ ጊዜ ባክቴሪያዎች በተለያየ ምክንያት ይሞታሉ። ሲሞቱ የነሱ ጄኔቲክ ማቴሪያል ኢንቫይሮንመንት ላይ ፍሎት እያደረገ ሊሆን ይችላል። ወተር ውስጥ ፍሎት እያደረገ ሊሆን ይችላል። ያ ባክቴሪያ ከዛ ከሞተው ባክቴሪያ ወይም ከተሰባበረው ባክቴሪያ ውስጥ ያለውን ጄኔቲክ ማቴሪያል እዛ ኢንቫይሮንመንት ላይ ፍሎት ከሚያደርገው ከሚንሳፈፈው እዛ አካባቢ ላይ ካለው አብዞርብ ሊያደርግ ይችላል። በኢንዶሳይቶሲስ ፕሮሰስ ምን ሊያደርግ ይችላል? ቴክን ሊያደርግ ይችላል። በዚህ መንገድ ከኢንቫይሮንመንት ላይ ያለው ፍሪ አብዞርብ ካደረገ ይሄ ምን ይባላል? ትራንስፎርሜሽን ይባላል ማለት ነው። እነዚህ ሜካኒዝም ጄኔቲክ ኤክስቼንጅ ሜካኒዝም፣ ኮንጁጌሽን፣ ትራንስዳክሽን ኤንድ ትራንስፎርሜሽን። በእነዚህ መንገድ ባክቴሪያው ምን አቴን ያደርጋል? ጄኔቲክ ዳይቨርሲቲ። ነገር ግን ስሩ ባይነሪ ፊዥን ኦሪጂናሉ ወይም ፓረንቱ ከዳውተሩ ጋር በምንም አይለያይም። አይደንቲካል ነው። ሌላው ወደ ቀጣዩ ዶሜን ኦፍ ላይፍ ነው የምንመጣው። አርካያ። እነዚህ አርካያ የሚባሉት የባክቴሪያል ግሩፖች ምን አይነት ናቸው? they are known to live in extreme environments. Okay, they live in extreme environments. There are three groups. Okay, there are of three groups like thermophiles, methanogens and halophiles. We will discuss about them. First, let's discuss about the characteristics of archaea. Okay, they have unique cell structures. They lack true peptidoglycans. Unlike that of the Eubacteria, they don't have true peptidoglycans in their cell wall. Their peptidoglycan is different from the peptidoglycan of what? Eubacteria. So they have different peptidoglycan. Actually, we can say that they have false peptidoglycan. They lack true peptidoglycan. And there is branched hydrocarbon chains in their peptidoglycans. Okay, they have branched hydrocarbon chains. They live in extreme habitats. For example, they may live in areas where it could be very difficult for different living organisms to survive. For example, in an area where the temperature is very high, some archaeas live. Such areas are known as thermophilic areas. Okay, thermophilies, extreme thermophilies. So such archaeas are known as extreme thermophilies, which live in extremely high temperature areas. Okay, in an area where the temperature is very high, which is usually above 90 degrees centigrade or above 80 degrees centigrade, which is very difficult for other living organisms to survive. So such bacteria are known as what? Extreme thermophils, okay? Or we call them thermophils simply. Some others live in an area where there is high concentration of salt or salt. As you know, salt has high effects on our body. If its content gets too much high, it has osmotic effect. It can cause water to be lost from our cells and can result in shrinking of our cell, and it may result in death of our cells and this may result in death of the entire the whole organism. Okay, but these archaea have like an adaptive feature that enables them to survive in a such area where there is high concentration of salt. Such archaea are known as hollow pills. Others live in an area where the pH level is very low. The pH where the pH is about 1 or 2, very low pH, which is acidic environment. Acidic environment is very difficult for many organisms to survive. But such archaea can live, and such archaea are known as acidopils. Okay, acidopils. But basically, we have 3 types of archaea based on their physiology. We classify them into 3 groups. Methanogens, those archaea that live in an area where there is no oxygen and which have ability to generate methane from degradation of organic matter. Since they have ability to generate methane, they are known as methanogens. They live in anaerobic area. For example, in waterlogged area, ውሃ ባቆረበት አካባቢ ውስጥ ላይ ኦክሲጅን ውስጥ ላይ መግባት የማይችልበት ውስጥ አካባቢ ላይ እዛ ይራባል። ውስጥ የሚገኝ ብስባሽ ነገሮችን በማበስበስ እነዚህ ኦርጋኒክ ሰብስታንሶችን ባዮዲግሬደብል የሆኑትን በማበስበስ ከዛ ሜቴን ጀነሬት ያደርጋሉ። ለምሳሌ የከብትበት ላይ ዳሪ ላይ ባዮጋዝ ጀነሬተር ውስጥ እዛ ውስጥ እነዚህ ባክቴሪያዎችና አርኪያዎች ናቸው ማለት ነው ስራው የሚሰሩት ምን በማድረግ ሜቴን ጋዝን ጀነሬት በማድረግ። ሌሎችም አሉ በጣም ኮልድ ኤሪያ ላይ የሚኖሩ በጣም ኮልድ ኤሪያ ላይ ሳይክሮፓይልስ የሚባሉ አሉ። ሳይክሮፓይልስ በጣም አንታርክቲክ ሪጅን ላይ በጣም ኮልድ ቴምፕሬቸሩ ከ4 ዲግሪ ሴንቲግሬድ በታች የሆነበት ፍሪዚንግ ኢንቫይሮንመንት ላይ የሚኖሩ አሉ። ሳይክሮፓይልስ ይባላሉ። ወይም ካይሮፓይልስ ክሪዮፓይልስ ነው የሚባሉት። ክሪዮፓይልስ ሌላው ኢምፖርታንት ካራክተሪስቲክሳቸው ምንድናቸው? ፕሮካሪዮቲክ ናቸው። ልክ እንደ ኢዩባክቴሪያ ፕሮካሪዮቲክ ናቸው። ዘ ኤላክትሮ ኒኩላር መምብሬን የላቸውም። ያ ማለት ጄኔቲክ ማቴሪያሎቹ ሲምፕሊ ሳስፔንድ ነው የሚያደርጉ ሳይቶፕላዝም ላይ። ይሄ ለባክቴሪያም ጭምር ነው። ለኢዩባክቴሪያ ጄኔቲክ ማቴሪያሎቹ ተሸፍኖ አይደለም ያለው በመምብሬን ወይም በሽፋን ተሸፍኖ አይደለም ያለው። በኖርማል ኢኳሪዮቲኮች ላይ መምብሬን ባውንድድ የሆነ ኒኩለስ ነው። ያ ማለት ጄኔቲክ ማቴሪያላቸው በደብል መምብሬን ምን ሆነ ተከቦ ነው ያለው። ያ ኒኩላር መምብሬን ይባላል ሽፋኑ። እነሱ ግን የላቸውም። ሲምፕሊ ሳስፔንድ ነው የሚያደርግ በሳይቶፕላዝም ላይ ነው የምናገኘው። ያ ኤራ ኒኩሎይድ ሪጅን ነው የሚባለው ብዙ ጊዜ ያ ጄኔቲክ ማቴሪያሉ ሳይሸፈን የሚገኝበት ሪጅን። ሶ they are single cell organisms አንድ ሴል ብቻ ናቸው። ኦርጋኒዝሙ አንዱ ሙሉ ኦርጋኒዝም አንድ ሴል ብቻ ነው ያለው without tree nucleus. Archaea represent a distinct domain of life separate from bacteria despite their similar appearance. Their unique adaptations allow them to survive in environments that would be lethal. Lethal means ገዳይ ነው ለሌላው ገዳይ ሊሆን የሚችልበት እነሱ ግን መኖር ይችላሉ ማለት makes them fascinating subjects for scientific study. So we have already mentioned there are major groups of archaea like methanogens. These anaerobic organisms that produce methane as a metabolic byproduct. They are commonly found in oxygen poor environments. They are anaerobic, like in marshes, swamps, animal intestines, playing a crucial role in carbon cycling. ስለዚህ በአንዳንድ በእንስሳቶች አንጀታቸው ውስጥ ጭምር እዛው ይለው ለምሳሌ አንዳንድ ከብቶች ሚቴን ጋዝ ያወጣሉ አይደል? ሩሚናንስ የሚባሉ እነዚያ መንጃጌዎች አፋቸው ሚቴን ጋዝ ይወጣል። ያ የሚወጣው ለምንድን ነው? እነዚህ ባክቴሪያዎች በአንጀታቸው ውስጥ ሆነው የበለጡን ኦርጋኒክ ማቴሪያል በመሰባበር በኦክሲጅን በሌለበት ኢንቫይሮንመንት ሚቴን ጋዝ ጀነሬት እንዲሆን ያደርጋሉ ማለት ነው። ኤክስትሪም ሃሎፒልስ የምንላቸዋል። እነዚህ ደግሞ ኦክሲጅን ባለበት ነው የሚኖሩት፣ ነገር ግን ከፍተኛ የሆነ ጨው ክምችት ባለበት። So they are salt loving archaea ናቸው that thrive in high salinity environments. የት አካባቢ? ለምሳሌ በጣም ጨው የበዛበት ባህር ላይ፣ ሐይቅ ላይ፣ great salt lakes, dead sea ህይወት የሌለበት ባህር ላይ እዛ በጣም ጨው ክምችት ያለበት። Many display distinctive red purple pigmentations that protect them from intense sunlight. Extreme thermophiles, heat loving archaea that are found that flourish in scorching environments like volcanic vents. In a volcanic eruption በጣም ከፍተኛ ቴምፕሬቸር ያለበት እዛ አካባቢ ይኖራሉ ማለት ነው ከ80 ዲግሪ ሴንቲግሬድ በላይ። their heat stable enzymes have valuable applications in biotechnology. በባዮቴክኖሎጂ ደረጃ እነዚህ አርካያዎች በጣም ፋንክሽን አላቸው፣ ኢምፖርታንት ሮል አላቸው፣ በጣም ጠቃሚ ናቸው። ለምሳሌ በምን ይጠቅማሉ? ኢንዛይማቸው ከነሱ ኤክስትራክት የሚሆን ኢንዛይም ለምሳሌ ፕሮቴዝ ኢንዛይም ከባክቴሪያ ሊገኝ ይችላል። ከዛ ባክቴሪያ ከሴል ውስጥ የሚገኘው ኢንዛይም ኢንትራሴሉላር ሊሆን ይችላል፣ ኤክስትራሴሉላር ሊሆን ይችላል። እንደሚታወቀው ኢንዛይም ባክቴሪያዎች ወደ ውጪ ሊመገቡ ብለው ኤክስትራሴሉላር አድርገው ውጪ ላይ ያለው ሊያፈራርሱ ይችላሉ። ሳፕሮፓቲክ ኒትሪሽን ላላቸው ለፋንጌዎች ነው ቦር የሚሰራው። ግን እነዚህ ኢንዛይሙ ከሴል ውጪ ፕሮች ሊሆን ይችላል፣ በሴል ውስጥ ፕሮች ሊሆን ይችላል፣ ኢንትራሴሉላር እና ኤክስትራሴሉላር። ለምሳሌ ኢንዛይም እንደሚታወቀው ምንድነው? ፕሮቲንን የያዘ ነው። ኢንዛይሙ ፕሮቲን የሚሰባብር ኢንዛይም ሊሆን ይችላል። ኢንዛይሙ ሊፒድ የሚሰባብር ሊሆን ይችላል። ፕሮቴዝ ኢንዛይም፣ ሊፔዝ ኢንዛይም፣ አማይሌዝ ኢንዛይም ሊኖር ይችላል። እና ለምሳሌ ብዙ ጊዜ ከነሱ ኤክስትራክት የሚሆነው ኢንዛይም ስፔሻሊ ከተርሞፒሊክ ከሆኑ ወይም ከተርሞፒልስ ከሆኑ አርካያዎች ኤክስትራክት የሚደረገው ኢንዛይም ለምን ሊጠቀሙት ይችላሉ? they may use it as in association with detergents in washing powders ጋር አንድ ላይ አድርገው ስቴኖችን ቆሻሻን ከልብስ ላይ ለማስወገድ አንዳንድ ለምሳሌ ቅባት የሆኑ ነገር ፕሮቲን ያላቸው ነገሮች ስንመገብ ልብሳችን ላይ ፈሶ ከሆነ እንቁላል አይነት ነገር ፈሶ ከሆነ ፕሮቲንነት ያላቸው ነገሮች ቆሻሻ ሆኖ ልብሳችን ላይ ካለ ያንን ቆሻሻ ከልብሳችን ላይ ለማስወገድ ፕሮቴዙ ኢንዛይሙ በቀላሉ ዲግሬድ አድርጎ ፕሮቲን ሰባብሮት ከልብሳችን እንዲወገድ ከዋሺንግ ዲተርጀንቱ ጋር አብሮ ተቀላቅሎ ያገለግል ይችላል። በምሳሌ በላውንደሪ ላይ በጣም ሃይ ቴምፕሬቸር በምንጠቀምበት ሰዓት ያ ኢንዛይም ብዙ ጊዜ ቴምፕሬቸር አይቋቋምም ብዙ ጊዜ ኢንዛይሞች። ነገር ግን ከነዚህ የሚገኙ ስፔሻል ነው። ለምን? ባክቴሪያዎቹ እነዚህ አርካያዎቹ ኤክስትሪም ቴምፕሬቸር ባለበት የሚኖሩ ስለሆነ ኢንዛይሙም አዳፕቴሽን አለውና ይጠቅማል። Enzymes derived from thermophilic archaea are used in detergents to function effectively at high temperatures and pH levels. These biological catalysts maintain their structure and function under conditions that would be denature most proteins. Okay? በዚህ መንገድ ቆሻሻዎችን ልብሳ የፈሰሰ ግሪስ ነገር ካለ ልብሳችን ላይ እነዚህ ሊፔዝ ኢንዛይሞች ተጠቅመው አንድ ላይ አድርገው ከቆሻሻዎች ስቴኖችን ያስወግዳሉ ማለት ነው። ስለዚህ በዚህ ደረጃ ይጠቅማል። ሌላው ባዮሪሚዲሽን ፖቴንሻል ለአርካያዎቹ አካባቢ ላይ ያለው ቆሻሻዎችን በማስወገድ ደረጃ ትልቅ ሚና አላቸው። አካባቢ ላይ ያለው ቆሻሻን እነዚህ ዲግሬድ እንዲያደርጉ እነዚህ አርካያዎቹ ቢጨመሩ ምን ኦክሲጅን በሌለበት ኢንቫይሮንመንት ያንን በመሰባበር ሚቴን ጋዝን ጀነሬት ያደርጉልናል። ለምሳሌ አካባቢ ላይ ዝም ብሎ ቢጣል በጣም አካባቢን ሊያገማ ይችላል፣ ሊያበላሽ ይችላል። ነገር ግን ያንን እንደ ባዮጋዝ ማብላያ ውስጥ ጨምረው አርካያዎቹን ከዛም ሚቴን ጋዝ እንዲገኝ ይችላል። አንዳንድ ደግሞ ዌስት የሆኑ ነገር ግን ፕሮቲን በውስጣቸው የያዙ የእንስሳት የሞቱ እንስሳት ሊሆኑ ይችላሉ፣ አካባቢ እንዳያገቡ ይሰባብራሉ ማለት ነው። So archaea's ability to thrive in extreme environments makes them promising candidates for what? Cleaning up contaminated sites. በቆሻሻ የተሞሉ አካባቢዎችን ማጽጃ ሆኖ ያገለግላሉ ማለት ነው። ምክንያቱም ስለሚሰባብር አካባቢ ላይ አሁን የሞተ ኦርጋኒዝም እዛም ሞቶ እንዳይኖር ከሚያደርጉት አንዱ ነገር ምንድነው? ያንን ሰውነት ላይ ያለውን ነገር የሚሰባብሩ ማይክሮ ኦርጋኒዝሞች አሉ። ከነዛ መካከል አርካያ ትልቅ ሚና ይጫወታል። So their unique metabolic pathways can break down pollutants that other organisms cannot process. በእንደዛ አይነት harsh environmental condition ውስጥ የሚሆንበት ሰዓት ሌሎች ማይክሮ ኦርጋኒዝም መስራት ማይችሉት መሰባበር ማይችሉትን እነዚህ ይሰባብራሉ። ሌላው በፖሊመሬዝ ቼን ሪአክሽን ቴክኖሎጂ ነው። the discovery of thermus aquatus a thermophilic archaeon revolutionized molecular biology through polymerase chain reaction. Its heat stable DNA polymerase enzyme is crucial for DNA amplification technique used in research and diagnosis. ለምሳሌ ለአንዳንድ ጊዜ ለሪሰርች እና ለዲያግኖስቲክ ፐርፐዝ ለምሳሌ ፎረንሲክ ቴክኖሎጂ ላይ የተገኘው ዲኤንኤ ሳምፕል በቂ ሆኖ ላይገኝ ይችላል። ለምሳሌ አንድ ሰው ወንጀል ቢሰራ እዛ አካባቢ ላይ የተገኘው ወንጀል ፈጻሚ ነው ተብሎ የታሰበ ሰው የዲኤንኤ ናሙና የተገኘው ደም ላይ ያለው ዲኤንኤ ናሙና አነስተኛ ሆኖ ሊገኝ ይችላል። ያንን አምፕሊፋይ አድርገው ዲኤንኤ ማልቲፕላይ አምፕሊፋይ የሚያደርጉበት ቴክኒክ ፖሊመሬዝ ቼን ሪአክሽን ይባላልና በዛ ቴክኒክ ላይ ይሄንን ኢንዛይም ከዚህ ከአርካያ የተገኘው መጠቀም ይቻላል ማለት ነው። በጣም ከፍተኛ የሆነ ሂትን ኢንቮልቭ ሊያደርግ ይችላልና በዛ ሁኔታ ውስጥ ያንን ኢንዛይም ተጠቅሞ ምን ሊያደርጉ ይችላሉ? ያንን አምፕሊፋይ ሊያደርጉ ይችላሉ ዲኤንኤው። Thank you for your attention, for joining me today as we explored the unseen yet powerful world of microorganisms, from the structure and the types of bacteria to the genetic structure and the types of bacteria to the genetic structure they use to survive and the unique qualities of archaea. I hope this session helped you learn something new. A special thanks to the Bright Technotonic company for their great support in making this video possible. ስለዚህ በጣም አመሰግናለሁ። በስተመጨረሻ አስተያየት ካላችሁ ኮሜንት ካላችሁ ማስቀመጥ ትችላላችሁ። ቀጣይ በሌሎች ኤሪያዎች ላይ ቪዲዮዎችን እናዘጋጃለን። ስለነበረን ቆይታ በጣም አመሰግናለሁ። መልካም ቆይታ